Surrounded by nature, plants produce defensive molecules in response to environmental attack. Each part of the plant produces specific molecules. Using this property, it is possible to force a plant to synthesize a molecule of interest. A stem cell is a non-specialized cell capable of self-renewal. It has the potential to become many types of specialized cells. It is called cell differentiation. Meristem cells are stem cells found in plants. They are located in the growing apical and root tips and are very difficult to isolate.
Only a small amount of tissue is required to make plant cell cultures. It can be a seed or a small piece of leaf that can be utilized to produce the plant cell cultures. And this is more sustainable for the environment and for the plant itself. This method has been utilized in IRB Bioderma to provide active substances for cosmetic applications. And it allows to utilize rare and endangered plants that otherwise would not be possible to use for commercial purposes. This process is particularly advantageous because the biological cycle is accelerated, and we can provide continuous and consistent supply of natural active ingredients according to the needs, and without being limited by the supply of or the harvest from natural sources. It is particularly interesting that we can use also slow-growing plants, as even plants living more than 100 years can be utilized.
At this stage, the dedifferentiated cells obtained can produce any part of the plant they are issued from to obtain a whole plant.
Thanks to exclusive method developed by IRB Bioderma, we can guarantee the supply of fully standardized ingredient and optimized in terms of active component presence. And this allows to have reproducible efficacy of the ingredient. Furthermore, the growth of cells in a perfectly sterile environment guarantees a 100% contaminant-free product, which is a safety guarantee for the consumer. Finally, the reproducibility of the cell line is also guaranteed by the fact that the same cell line is reutilized from batch to batch, and this can occur for a long period of time, as cell cultures can be utilized for more than 13 years.
The first step is to select the optimal protocol to get the best dedifferentiated cell line. Then the biomass is increased in order to improve the number of cells and thus their production capacity. This is the tightest part of the job, as it consists in setting up the optimum parameters, biological, biochemical and physical, to maximize the production of molecule of interest by the cells.
IRB Bioderma has developed several cell lines. More than 100 different species have been utilized, and have adapted several different protocols to maintain and grow these cell lines. This allows the possibility to provide the biggest callus cell bank available for industrial purposes. Furthermore, we have also developed a unique preservation method that allows to have a long-term storage of the cell cultures, and as well provides the possibility to register in a cell bank and have a patent coverage for the cell lines, and is a valuable promise for future active ingredient development.